I did my research paper on Gendicine, which is a gene therapy that was approved in 2003 for head and neck cancers, and this was approved in China by the country's State Food and Drug Administration, and it has been proved safe and effective and has also been applied to treat a variety of other cancers, but in 2003 it was only approved for head and neck squamous cell carcinoma. So Gendicine has been proved safe and effective, especially when paired with traditional cancer treatments like radiotherapy, chemotherapy, and hyperthermia. So this gene therapy was created by Cybiono Gene Tech, and it was approved to be administered through a minimally invasive injection, usually at the site of the tumor, or by intracavity or intravascular infusion. So the treatment involves recombinant adenovirus, which is modified to express the wild-type p53 gene. And the p53 gene does occur naturally during cell development, and it leads to it can either lead cells to apoptosis, which is important for tumor cells, or it can foster the survival of a cell in natural development. But it does lead to the implementation of tumor-suppressing abilities, and it goes into the nucleus of tumor cells. So the gene is seen as suitable for cancer treatments because it can halt the cell cycle and induce apoptosis, which is what Gendicine does when it is injected into a tumor. So it can lead to cell death of tumors, and it can get rid of cancer within a patient. But p53 is correlated, or like a low number of p53 within tumor cells is correlated with a lower rate of patient survival and a greater resistance to chemotherapy, and more than half of human cancers, p53 is the most mutated or deleted, regularly altered gene, so the gene does have a critical role in slowing cancer cell growth. And with Gendicine, the WT-p53 gene, which is the wild-type p53 gene, is restored in malignant tumors, and it introduces a copy of complementary DNA of the p53 gene using a vector of an adenovirus nature. So the p53 gene straight into the malignant tumor cells has displayed the suppression of cancerous growth because of its ectopic expression. So the externally derived gene reacts to its abnormal environment, and it recognizes that it's in a situation of stress, and it signals the activation of cell death, and then tumors can be killed within a patient. So adenoviruses are better suited for the gene therapy because vector DNA doesn't integrate into the host cells. So it does not replicate itself, but it does still lead to better patient cancer survival. And there was difficulties during clinical trials in getting effectiveness towards patient survival. At first, the adenovirus did produce small amounts of toxicity as well, but that was resolved. And during one clinical trial where there was a combo group receiving Gendicine and an infusion, a bronchial arterial infusion, there was that combo group, and then there was a control group that only received the infusion, and the combo group had a longer disease progression than that of the control group, but neither of them had better chances of survival. So gene therapy did allow the disease to progress more slow, but it didn't necessarily give them better chance of surviving the disease. So through more clinical studies and trials, the patients' survival rates did improve. So in 2005, there was a clinical study in which a young patient with hepatocellular carcinoma received combination treatment with Gendicine and a transcatheter arterial chemoembolization. And 4 days after the procedure, the patient's hepatic arteries were embolized, and 7 months later they were in good health with a normally functioning liver. So it did get rid of the cancer inside of him. So 150 patients were involved in a clinical study in China, and 68 patients were treated with a combination of TACE And Gendicine, while 82 patients received only the TACE treatment, which is the transcatheter arterial chemoembolization, and the combo group that received Gendicine had a 6-month survival rate of 76.5%, but the control group had a survival rate of only 23.2%, so Gendicine greatly improved their survival rate. And it does show that Gendicine has huge potential benefits when combined with several different treatments. So there was also a study in which patients were being treated with HCC, or they were being treated for HCC, and all the groups that received the combination of traditional cancer treatments along with Gendicine had improved survival rates. So it is common for patients to have a better survival rate when treated with Gendicine and any traditional cancer treatment. So there was years and years of research, as Gendicine was created 20 years after the first gene therapy trial, and it has proved itself as very effective for improving survival rates in patients with carcinoma.